Well, influenza is an important disease. It's a respiratory disease, and it kills approximately 60,000 Americans per year, and maybe a couple of 100,000 people throughout the world. And the 1918-1919 influenza killed about 50 million people. It was the greatest disaster known to mankind that has ever occurred, and in a short time reduced the population significantly, greater than any other illness or tragedy or nature disaster. And so our interest has been to try to understand influenza, how it works, and how to treat it, and how to prevent the effects from people dying from influenza. So my colleague, Hugh Rosen, has developed some medicinal compounds that have been extraordinarily effective in the treatment of influenza.
When the virus infects patients, there is a subset of patients who respond to the virus with a very aggressive immune response. This leads to lung damage, and these patients do particularly badly. So we hypothesized that treating the immunopathology, treating the immune response and blunting it would protect potentially the patient from infection while allowing the infected person to respond adequately to the infection and eradicate the virus. And using these molecules that are agonists of a receptor called S1P1, a receptor for a signaling lipid called sphingosine 1-phosphate, we were able to show that we could protect animals, enhance their survival in the face of infection with a pandemic human influenza isolate.
Correct. And one of the interesting parts is that influenza is caused by a virus, and it has been long thought that the virus per se is the culprit that causes the disease, but is important for the increased morbidity and mortality. And our work really is one that clearly shows that in certain patients that it's the immune response going awry against the virus that's the culprit, and that in fact by giving this type of therapy, which is against a host response, is as effective, in fact is more significantly effective than giving the current antiviral therapies that are on the market currently. And interestingly, by giving both in combination, the antiviral therapy and the therapy against the sphingosine 1-phosphate 1 receptor, at least in animal studies, I give the maximum protection that's observed.
In addition, targeting the host response allows us to target something that doesn't evolve and mutate as rapidly as the virus, and so one has a much diminished chance of generating resistance to the therapeutic approach. So these basic science experiments, which in fact are designed to delineate basic mechanism and to define new therapeutic targets, provide the first direct evidence for amelioration of the host response as a means of providing protection from pandemic strains of influenza, and this can be achieved chemically with the types of small organic molecules that can be well tolerated as drugs. And so further work now needs to be done to develop this for assessing it in human patients over time.
And I suppose the other point that really should be made is the synergy between our two laboratories in doing this, which provides a foundation of what Scripps is really about, that allows scientists with interests, mutual interests, to work together with no boundaries and to work on important problems. And we hope to come up to very essential findings that will be helpful in terms of treating human disease.
Well said.